Thanks for joining us today. We're very honoured to have Sheila Peters join us to give a talk based on the recent IQ paper. So it's focused on the organ impairment populations in SimSip. Just to let you know, you'll probably be muted on the line, but if you have any questions, please use the Q&A or the chat features, and I'll make sure either your questions read out or you're unmuted so you can ask your question. If you go on to the next slide, Sheila. Just to mention as well that this session is recorded and will be available on YouTube afterwards. So just a bit about Sheila for those of you who don't know Sheila. She originally did her PhD in chemistry in India and started her career in R&D in working in Cyprotex. So I've known her through AstraZeneca, and she developed her expertise there in PBPK modeling. She built her own model in MATLAB. And since then, she's moved to Merck, and she's head of translation and quantitative pharmacology group. She's active not just within Merck, externally. She is part of the ICH group looking at harmonizing the DDI guidelines. She's going to be talking a bit about the paper related to IQ work, and also she's attended EMA organized meetings. So perhaps that was before Brexit, because it looks like you may have met in London. She's served on, she's published several papers in PK and PBPK. She's an expert in this field. We've collaborated on some gut metabolism papers, and she's also published on drug-drug interactions. And if you didn't know, she also has a book on PBPK, so she's an expert in this field. So today, without further ado, I'll pass over to Sheila to give a talk on the IQ paper on organ-impaired populations. So thanks again.
Thank you, Oliver, for a very kind introduction. And as Oliver pointed out, I will talk to you today about the IQ working group evaluation of PBPK models for organ-impaired populations. The working group members are drawn from 19 member companies under the Translational and ADME Science Leadership Group, as well as the Clinical Pharmacology Leadership Groups, arms of the IQ Consortium, shown here. Before I could start with the presentation, I would like to say that work that I will talk about is published in the CPT last year as a white paper. Before I go to the objectives of our work and the evaluation, I would also like to talk about some pathophysiological changes in patients in organ impairment, both for renal as well as for hepatic. First, starting with the renal impairment. Renal impairment is characterized by a reduced glomerular filtration rate and changes in the kidney size as well as gastric emptying time. It is also characterized by accumulation of uremic toxins. And the accumulation of uremic toxins in turn can lead to a reduced CYP activity, either by downregulation or by the direct inhibition of CYP enzymes. It can also lead to inhibition of transporters like OAT. Uremic toxins can increase the unbound fraction of drugs by competitively binding to plasma proteins or by inducing hyperalbuminemia, at least in some patients. Thus, renal impairment, through altering the CYP activity and the transporter activity and binding to plasma proteins, can even affect the exposure of a drug that is not primarily eliminated by the kidney. We have used SimSip version 15 in our evaluation, and SimSip, this version captures the physiological changes in glomerular filtration rate, CYP abundance, albumin, kidney size, and gastric emptying time, all of the features that we had pointed out here. However, the inhibition of enzymes and transporters are not considered in a mechanistic way. I was just talking to Oliver a few minutes ago, and he said that it is accounted for in simply a reduction in the CYP activity. But this is perhaps something they can improve in the future. Now, coming to pathophysiological changes in patients with liver cirrhosis is characterized by a progressive decline in the hepatic blood flow, hematocrit, alpha 1 glycoprotein, albumin, functional hepatocytes, and biliary excretion. It is also characterized by alterations in the expression and activity of hepatic transporters and metabolizing enzymes, as well as duodenal CYP3A. It is, cirrhotic patients also have insertion of portocaval shunt. And this is illustrated in the figure on the right, where you can see a normal liver and a cirrhotic liver. In the cirrhotic liver, the formation of connective tissues and nodules in the liver can obstruct the flow of venous blood. And to overcome this intrahepatic pressure, there is an insertion of the portocaval shunt here, as you can see, to relieve the interhepatic pressure. So this insertion of the portocaval shunt is a compensatory mechanism. And with disease progression, this compensatory mechanism can become inadequate. And therefore, the effective arterial blood volume declines eventually, leading to a diminished renal blood flow, which in turn triggers the renin-angiotensin aldosterone system, or the RAS system. And this in turn leads to the resulting hyperdynamic circulation, which leads to a reduced glomerular filtration rate, and finally to an acute renal failure. Thus, again, like previously, we had seen that the liver impairment can impact even drug exposure of hepatically eliminated drugs. Here we can see that the liver disease can impact the kidney function and alter drug exposure, for drugs that are significantly eliminated renally. SimCip version 15 captures the pathophysiological changes in renal and hepatic blood flows, gastric emptying time, glomerular filtration rate, CYP abundance, albumin, alpha 1 acid glycoprotein, hematocrit, and liver volume. Additionally, there has been a recent publication from the University of Manchester that calls for likely changes in the scalars that are used. So even though the volume is reduced, for every 1 gram of that volume that is available, there could be a further reduction in the milligram protein. And therefore, that is something that is not currently available in SimCip. So now going to the IQ Consortium evaluation of organ impairment PBPK models. Before I go to that, I would like to just briefly go through with you the regulatory recommendations of studies in organ impaired populations. Organ impairment mediated drug accumulation can impact drug safety, and PK studies are therefore recommended by the regulatory agencies for drugs that are likely to be used by the organ impaired patients and primarily eliminated by the impaired organ. And this study must be conducted in patients of varying disease severity in a full study. And for the renal impairment, this disease severity is measured in terms of estimated glomerular filtration rate or creatinine clearance. For the hepatic impairment, the disease severity is in terms of the child-poor classification as mild, which is child-poor class A, and B is moderate, and C is severe. Now, there is a huge opportunity for PBPK modeling because these studies are quite resource intensive. It can take several years to recruit enough patients because we need at least 8 patients and 8 in the healthy controls. And we need, for the full study, patients from different disease severity. And so it takes quite a long time. And also, therefore, it will also cost a lot. And therefore, there is a great opportunity for PBPK modeling of organ impaired populations to supplement limited data that could be derived from fewer patients, if to reduce the timelines and cost, if the models are validated for this purpose. And Unfortunately, today, the regulatory confidence in the PBPK models for organ impaired populations is low. And this is because the validation that we talked about here is not really there. There are isolated examples that have been reported of PBPK models for different organ impaired patients, both liver and in renal. But these studies are in very few compounds. And also, they are not done in a standard way, and therefore the conclusions one can draw from them cannot be very firm, and therefore there is not so much confidence today. So that was the kind of a motivation for us to evaluate the PBPK models for OI populations using standardized workflow on a large data set with a range of clearance and elimination pathways. And the question we wanted to answer with this evaluation is the performance of the OI PBPK models then are sufficient to rationalize the need, timing, and design of OI studies? Or can they be used to supplement the limited clinical data when recruitment is incomplete? As I said before, these can take a long time, and therefore, if it is incomplete, perhaps you can use the sparse data and supplement that with the model predictions. Or sometimes it is even difficult to get these patients. Sorry, it's even difficult to get these patients if it is, for example, in children or in oncology settings. So in order to support this goal, we collected data, large data set over the last 5 years. And, the data was coming from 19 IQ consortium member companies for 29 compounds. And for these 29 compounds, there were 56 clinical study arms for hepatic impairment and 50 clinical study arms for the renal impairment. Now you can see the distribution of these study arms in the various categories that we mentioned here for the renal impairment and hepatic impairment. That is a mild, moderate, and severe based on the different, parameters that were shown there. And for the renal impairment, you can see in the same way the distribution of patients in these 50 RI study arms were not bad for the mild, moderate, and severe. But for the end stage renal disease, it was very small number. So there was not much that we could help here. We could only get about 3 end stage renal disease. But otherwise, the distribution was quite evenly spread on between the different disease severity groups. So now going to the standardized workflow that we followed for the evaluation. So the model was built, the base PK model for the drug was built using physicochemical properties and in vitro data, clinical PK data coming from first in human single ascending dose, and also pathway contributions like FM-CYP, fraction eliminated in urine or in bile. And if mass balance data was available, we used that for the pathway contributions as well. And once these models were built, they were verified and refined. These base PK models were in the healthy were verified and refined. And these verifications were supported by multiple ascending dose data or DDI studies in healthy volunteers or patients. The patients here that I talk about are not hepatic impaired or renal impaired patients, but these are, for example, in oncology, we won't be able to do these studies in healthy volunteers. So in those cases, we will have the patients for drugs that cannot be administered to healthy volunteers. So that is what the patients here refer to. It's not the organ impaired patients. So once we have a base PK model verified and defined, we use that to predict the PK of the healthy controls. Because in every organ impairment study, we have the healthy controls. So we predicted the PK of the healthy controls in those studies. And once we had a satisfactory prediction, and that means that the predicted exposures in the healthy controls were within twofold of the observed. And when this condition was met, then we went on to build the PBPK model for the organ impairment by including the OI organ impairment specific parameters like blood flow, glomerular filtration rate, CYP abundance, all of the features that I earlier alluded to. Now, on top of this, we can also do a sensitivity analysis to build the robustness and confidence in the model. So mainly for parameters where we have some kind of uncertainty, perhaps in the plasma protein binding data, because if the ex vivo measurements are not available, then we use a predicted plasma protein binding in the impaired population, and this can be then subject to uncertainty analysis. Once we are convinced of the robustness and we are confident about the OI PBPK model, can then use it to simulate the PK in the mild, moderate, and the severe OI patients, whether it is renal or hepatic. The workflow was the same. Now, all models were built in SimCip version 15 and validated by modelers from the contributing companies. Human ADME data were available for most of the compounds, 75% of the compounds. And if a compound was metabolized by enzymes with known genetic polymorphism, then simulations were also performed in subjects with different metabolic status. For example, for CYP2D6, 2C9, 2C19, we also did the simulations for the different metabolic status subjects as part of verification. For the organ-impaired models, ex vivo measured protein data were used when available, as I said before. Otherwise, it was predicted by the SimCip model. Virtual trial simulations, 10 trials with 10 subjects were performed, and with matched clinical study design with respect to dose, dosing frequency, age, range, and gender ratio of the study cohorts. Prediction success was assessed using twofold prediction error and the guessed criterion. Now, coming to the results. Now, this slide shows the AUC ratios of renal impaired patients to matched healthy controls, as well as the Cmax ratios in the renal impaired patients to matched healthy controls. So here the AUC ratio refers to the exposure in the impaired patients, organ impaired patients, to the exposure in the healthy controls. So if you expect to see in an impaired population and higher exposure, that means your AUC ratio would be greater than 1. So I would focus on the AUC ratio, and as you can see, the results are very similar. Of course, there is this one exception, and I'll come to it in a minute. So the first of all, let's see how the observed effects were seen. You can see that the observed effects were, the range was quite modest. So it only covered between 0.6 to 2.2. So it's quite a narrow range, around 0.6 to 2.2. And the only exception there is this compound. And this compound is actually a metabolite of one of the drugs that were studied here. So this metabolite is probably using the renal pathway to a much larger extent than all of these parent drugs. And therefore, the magnitude of effect, which you can imagine, should be proportional to the extent of renal elimination for that compound, is quite high for this metabolite. Otherwise, the range is quite limited. And what you can also see here is the dotted lines represent the prediction error of twofold. And the red lines here are the guessed criterion. So as I said, we assess the models by both criteria, both by the twofold, by the dotted, as well as the guessed criterion. So the PBPK predictions were within the twofold for greater than 90%. So it was only these two that were outside the prediction error of twofold. And the accuracy of predictions was less than twofold for drugs for which the renal clearance contributed significantly. So in fact, it's these drugs which are really interesting to us, because these are the ones that for which the renal impairment study has to be done. And for this, the prediction is even lesser than twofold. And there is also a tendency to over-predict the effects of renal impairment on PK, drug PK, especially in groups with high disease severity. For example, these outliers here, both with respect to the twofold as well as with respect to the guess criteria, most of these are just over predictions. Only one of them, and this also is within twofold error, but outside of the guess criterion. But mostly it is over prediction, as you can see. And this over prediction is much more pronounced for the square and the cross, which is the severe and the ESRD. For the mild, which is the circle and the triangle, red triangles, which are moderate or RI, the predictions are very good. So as I said, similar trend was seen also in the Cmax ratio. Now, this is for the AUC ratios of the liver impaired patients to matched healthy controls. Now, for the liver, the disease severity classification is based on the Child-Pugh A, B, and C. So CPA is Child-Pugh class A, CPB Child-Pugh class B, and so on. So again, focusing now on the observed AUC ratios first, what you can see here is the range depends, of course, on whether we are talking about the CPA, CPB, or CPC. In the class A, the range is quite narrow, but when you go to higher disease severity, then you can see that the range is spread out. You still see quite a lot of compounds around one, even with a very severe case. And you have even values less than one. And I will come to why this can be the reason. And what you see here is that the PBPK predictions were within twofold of the observed for 70% of the study. So it is not as good as for the renal impairment. Here, of course, because you have a huge range as well to predict. And so the predictions were within twofold only for about 70% of the studies. And one thing to note here is absolutely there is no under predictions, not even one. Even with respect to the twofold, there is nothing. There we had seen, at least with the guest criterion, there was one outlier that was coming between the guest criterion and the twofold. But here in the liver impaired, there's absolutely no under predictions. Everything, there is a tendency, there is a systematic tendency for over prediction. So this is an important point to note because it is quite useful for us when we think about the applications of the model. So the CPA model appeared to have the best performance with only one outside the twofold error range. However, you can see here that when the guest criterion is applied, it's more than 50%, outside of the twofold or the upper limit. For the compounds with systemic clearance greater than 20 liter per hour, yeah, I forgot to mention here that the closed symbols represent clearance values less than 20 liters per hour. So all of these, you can see all the over predictions are coming for the closed ones, which are all low clearance, which is less than 20 liter per hour in our definition. So here again, you can see that they're all closed symbols, less than 20 liter per hour. And there is only one exception to that, and this is this compound. So now, again, what you can also see is that the proportion of over predictions also depends upon the disease. So you can see that it's not only on the compound dependent clearance, but it's also the disease severity. So you can see that this is only 5% here, and this represents a 20% of the overall that is outside of the twofold. And here it is like 40% outside the twofold. So there is an increasing tendency of the proportion of molecule of predictions outside of the twofold. So for compounds with systemic clearance greater than 20 liters per hour, then the predictions are very good. So most of them come within the guest criteria. And it is like 83% of the predictions for CPC and 82% of the predictions for CPB within the guest criteria, which is really good. So it is only the low clearance compounds for which you expect lower values, lower magnitude of effects, and therefore lower AUC ratios for those ones. And also when, disease severity increases, that is where you have the most outliers. So for compounds that were studied clinically in more than one hepatic impairment category, then the effects of the moderate HI, that is in the middle one, right? So in the moderate, if they were predicted within twofold, then the same could be applied to the other two categories as well. So if it was predicted within twofold in the moderate, then you can also, what was seen was they were also well predicted within twofold in the severe as well as in the mild. And that means if we had the measurement, if the study was done using moderate, this provides us confidence, to extrapolate to other classes, to CPA and to CPC, because when it is well predicted in moderate, it is also well predicted in within twofold in the severe and in the mild. Now going to the Cmax ratios of liver impaired patients to matched healthy controls. Again, here for the CPA, CPB, and CPC, the trends are very similar, like what you had before. The only one exception here is this very peculiar outlier here. And this is only the case in the mild. It's not there in the moderate and severe. In fact, this compound is 96% CYP3A. And what really distinguishes this compound is the huge clearance. This is IV clearance. It is 61 liter per hour. And if it's orally administered, you can see that there could be quite a lot of gut metabolism. And we had seen that there is a reduction in the the duodenal CYP3A as well. So it could be something to do with the gut metabolism as well. Okay, now I want to discuss a little bit more about the outliers to understand what could explain some of the outliers that we have seen. So our evaluation identified a tendency to overpredict the effects of impairment for some compounds. And as we had seen, this could be compound related. Factors such as extent of clearance. So we had seen in the case of renal, it was a low clearance compounds that were not, predicted properly, or it was over predicted. And sometimes we could explain by the pathways. For example, in the previous slide we had seen that it is a CYP3A. When it's very high contribution from the CYP3A, we had generally seen a much higher magnitude of effect of the impairment on the PK. But this could not explain all of the outliers that we had seen. So there could be other disease related component to explaining the outlier. This is what we think. And maybe that the models predict linear changes in the effects of organ impairment while with disease progression, whereas in reality, the impairment effects may be less than proportional. And this can explain why we are over predicting in the severe, because in the severe, the disease impact doesn't seem to be much, whereas in our model, we seem to have some linear changes of impact of disease, and we are seeming to predict higher values than what was actually seen in this population. So maybe there is some kind of compensatory mechanism with disease severity. And what we saw here in this plot of observed AUC ratio of impaired to healthy versus disease severity going from mild, moderate to severe in the hepatic impairment population, what we see is actually there is a tendency to some kind of compensation. In fact, you find that for some there is even going down, some of these are actually going down. So there could be some compensatory mechanism kicking in with increased severe disease. Then this nonlinearity may be attributable to compensatory mechanism, as we had seen, or alternatively, it can also be explained by the use of childhood classification, which is actually based on disease biomarkers and are not really designed to estimate the impact of hepatic impairment on the ADME properties of the drugs. So as we know, the childhood classification is based on very much, disease characteristics and not anything to do with the hepatic function or the enzyme and transporter function. So perspectives and future directions. The variation in outcomes due to different analysts conducting the work cannot be excluded. So this is something to keep in mind, but this is somewhat mitigated. This risk of different outcomes coming from different analysts is somewhat mitigated by the use of the standardized workflow which we agreed ahead. The compounds in the study had very little transporter activity. Therefore, the impact of the reduction in transporter abundance and activity for transporter substrates could not be assessed. Some molecules showed significant changes with severity and others did not. So that was very weird, and we could not relate this just to the extent of clearance or pathway. So this is a little bit weird. Why for some compounds with severity we cannot explain a increased magnitude of effect. And maybe there is, as we saw before, there is a compensatory mechanisms, but we don't understand this today. So a better understanding of the sources of variability in observed outcomes is necessary to further improve predictions in the organ impaired population. So we really need to understand what is differentiating the different outcomes. What is the reason? So we are not able to understand that today. So even though predictions could be made, reasons for outliers were not always clear. And of course, this could be attributed to some known gaps in version 15, as was pointed out earlier, some of them being the potential inhibition of CYPs and transporters in uremic toxins in the renal impaired population, and the importance of using relevant disease relevant scalars in hepatic impaired population. For an additional factor could also be that for lipophilic compounds, which depend on bile solubilization for absorption, a reduced bile acid output from the liver in the impaired patients could actually lead to a reduced drug absorption. And this could explain why some of them have a less than one observed AUC ratio. So of course, the less than one observed AUC ratio could be also because of protein binding. But the protein binding changes are not so significant, and that alone cannot explain. So maybe there are additional factors like this, because most of the compounds we looked at were oral compounds, and this factor cannot be disregarded. Um, Again, the accurate staging of hepatic insufficiency based on more appropriate imaging and non-invasive biomarkers rather than the disease biomarkers in the child pool may be needed for improved prediction. So these are some of our perspectives and how we see the future directions for model development. So now, given these results from our evaluation, how can we apply PBPK models? Now, the FDA draft guidelines for renal impairment that was released last September recommends an early characterization of the effect of renal impairment on a drug's PK. And this early characterization of the renal impairment can be based on early clinical studies or on modeling and simulation methods like PBPK. So this is what they mention. And early characterization is needed to enable inclusion of the organ impaired population in the late phase trials, because normally these patients are excluded so as to not, because there could be variability in the study results, or these patients could be at risk of safety risks and so on. So they are generally excluded. But instead, if we can characterize them early, they can be included with or without dose adjustments in the late phase trials. And this is what we call broadening the eligibility criteria in the late phase trials. So our evaluation suggests that we are usually over predicting rather than under predicting. So this is something I had pointed out for both the renal as well as hepatic impairment. In fact, in the hepatic impairment, there was absolutely no, and not even one out of the 56 clinical study arms, not even one was under predicted. So this there is therefore a systematic tendency for the model to over predict. So for such a model, if it comes up with a prediction of an AUC ratio between 80 and 125%, it's the equivalence range. In that case, we can be pretty confident that given the characteristics of that particular compound, the predicted effects by the model, which is very modest, very small, is reliable, and therefore we can actually request a study waiver, especially for drugs with wide therapeutic index. Now, if a study has to be conducted for the narrow therapeutic index, then the timing of that study could be later rather than earlier, because we know that these people are not having so much effect of impairment because of the nature of the drug characteristics. And therefore, we can actually time it later, and we could even include them without dose adjustments in late phase trials. And these confirmatory trials in the narrow therapeutic index drugs can be designed in a lean way because they are confirmatory studies, and that's one way. So if we don't know whether the drug has got a narrow therapeutic index or not, you can still use PBPK model predictions for broadening the eligibility criteria of the renal impairment studies. Now, another application is supplementing limited clinical data. For example, in the hepatic impairment, we had seen that when the model predictions were good in the moderate class, then the predictions were also good in the other two classes. And this gives us the confidence when we have measured in one class of patients, then we can also extend it to the other two classes. Simulations can always contribute to a totality of evidence coming from mass balance, from DDI studies, and so on. So what would happen if the magnitude of effect predicted by the model is not so low? It's beyond this equivalence range. Again, it depends on the therapeutic area. For drugs in oncology, we can still request waiver if the magnitude of the change predicted is small compared to relative to the therapeutic index. Again, it can be used for timing this study if the magnitude is small relative to the therapeutic index. You can use PBPK models to optimize study design with respect to doses to be tested and with respect to dosing frequency as well. You can also use it for broadening eligibility criteria, same as before, but here you did not require dose adjustments because the magnitude predicted was small. But in this case, you may have to do the broadening of eligibility criteria with dose adjustments, maybe. And again, we can supplement limited clinical data when recruitment is incomplete, and also use it for a contribution to the totality of evidence. So these are some of the applications we see given the model evaluations we have. So I would like to end my presentation with some key take-home messages. Model predictions were within twofold of the observed data in greater than 90% of the studies in renal impaired, and that we had 50 clinical study arms, and greater than 70% of the studies in hepatic impairment in 56 clinical study arms. Those outside of the twofold error tended to be over predictions in patients with more severe impairments. Given the tendency for the PBPK models to over-predict rather than under-predict, when model predictions of organ impaired to healthy AUC ratios are between 0.8 to 1.25, our study supports restricting the organ impairment studies to narrow therapeutic drugs. In the absence of information on the therapeutic index of a drug, PBPK model can contribute to a totality of evidence alongside mass balance, absolute bioavailability, and DDI studies to support the inclusion of organ-impaired subjects in late clinical trials, late-stage clinical trials, with or without dose adjustments. When PBPK predictions of AUC ratios are outside the bioequivalence limits, model predictions are likely to represent a worst-case scenario and may be used to design the confirmatory studies. Confirmatory studies in organ-impaired patients may be performed with a lean design in moderate or severe patients, and then the outcome used to support the label. Such confirmatory studies may be considered later in the development phase to save cost rather than early. When impairment effects were predicted within twofold in moderate hepatic impairment, the effects of mild and or severe HI were also predicted within twofold regardless of their clearance. And this finding therefore supports the use of PBPK predictions to supplement limited clinical data when recruitment is incomplete in one or more categories of impairment, or to supplement limited observations when recruitment is difficult, as in the case of oncology and pediatric settings. So with that, I would like to acknowledge the contribution of all the working group members towards preparing these slides, and have been extremely useful in answering many of my questions, sometimes in the last minute before coming here. So really very thankful to the entire team. And thanks to you all for your attention. And I'm happy to take any further questions.
Thanks, Sheila. That was a really good talk. Is there anyone, sorry, online with any questions?
We don't have any questions at the moment. We've got one from Guan King. I'll unmute them now.
yeah. So the question is about whether there'll be an improvement with version 20. I guess from what we were speaking at the beginning, a lot of the improvements that we're hoping to implement are part of the version 21 wishlist project. So I guess that answers your question for you. So in that project, just to expand, what we're doing is so before we've incorporated, for example, SIP changes, we are reviewing that data based on latest available data, also looking at non-SIPs, so UGTs, esterases, for example, and also incorporating changes in transporters. I guess in your work, Sheila, were they all SIP substrates?
Yes, mostly substrates. There were no transporter substrates, unfortunately. And most of them were
UGTs or anything?
CYP3A. No, no. I mean, there were contributions from UGT, but nothing as, the major pathway or anything. Certainly, there were a few compounds with UGT contributions.
Mm-hmm.
Ollie, I've just sent you another question that's come from Nikunj.
Nikunj, maybe you can just unmute Nikunj and he can answer.
Just find him. I've got him. Nikunj.
Hi, yes, hi. Thank you, Sheila, for nice
You're
visit.
quite quiet, Nikunj.
I'm sorry, it might be. Can you hear me?
Just about.
Yeah, no problem. I will try.
And yeah, I was just asking, did you try and tease out the effect of hepatoportal shunt, um, or see whether the effect of hepatic impairment on clearance versus first pass? So,
Uh,
so the clearance versus the first pass extraction, did you try and tease out, or did you try to see if that could explain some of your current predictions?
yeah, we tried to do what all we could do was to see what we certainly saw was when the magnitude of effects is small, then we are not able to predict, right? And when the CYP3A is a major pathway, then I think it's mainly coming from the gut metabolism part as well. So somehow the magnitude of effects seems to be very, very high. And with increased disease severity, there is larger over prediction. So these were the only things we could come across. I mean, and I think despite having such a huge number of compounds, you would think it's quite a lot. It's all of the compounds, compound data we have on hepatic impairment and renal impairment. It's quite the largest one can have. But still, when you tried to look at correlations, it was difficult to find, given all the variables here. It's the protein binding, it's the the variables that is causing the disease impairment, and the extent of clearance and the pathway. So then it becomes quite a lot actually, and we don't have sufficient numbers to power any further conclusions than what we have talked about.
Yeah, thank you.
You've got another question from Stephen Hall. I'll just unmute him as well.
Thanks. I don't know whether he's
Hi,
putting
Sheila.
it to me or to Sheila.
Can you hear me?
Yes.
Oh, great. That was a very nice presentation. If I could just speak to the previous question, that was one of the reasons we split out the lower clearance, less than 20, and the higher clearance compounds, because the shunt would have been much more important for the high clearance compounds with the low bioavailability. And as you pointed out, that didn't seem to represent an area where predictions were worse. So also, the shunting in SimSip is present when the full PBPK model is implemented. and in a number of the examples we had, that was not the modeling approach that the particular company took. So the shunting part was not really well addressed, I think, in this study design. But the question I had was, I always felt as we were doing this, there were two studies happening. We were looking at the SimSip predictions, but we were also looking at how a cross-industry investigation like this can happen, and what the problems, the pitfalls of that approach is. And so I was wondering, if we started today from the beginning, would we do this differently? How could we, perhaps with the new version that's going to come out, what kind of things could we do perhaps to increase our confidence?
Okay, nice question. I think now that we know what could be the potential contributions to the variability in the outcomes, perhaps we can focus more, on designing the study, like you said, in a better way to be able to answer the questions we have. So I think this evaluation has provided us some directions that could be of help, and typically in the design of the standardized workflow.
You've got another question from Masood as well. Would it be possible to get compounds, workspaces to run them in the new version?
I think it should be, right, Steve? Do you know if it's all available in the IQ?
Well, I mean, one of the conditions of the IQ working groups is this kind of protection of the confidentiality, if you like. So the compounds are anonymized. None of the folks in the working group knew what they were. They were just given a code name by the IQ consortium. And then the results, the observed and predicted, could be compared. So it's not immediately obvious whether we can share them or not. We would have to talk with the IQ people, and they may have to then address that question with each of the companies involved. So it's something, I think we've probably had this conversation before. We cannot immediately share those, but possibly there's a path forward. We would have to take some advice on that.
And I think there's a comment from Taicho. Care should be taken for compounds where liver bile secretions impact absorption.
Yes, so this was also one of our, I think here, the conclusions that we, for lipophilic compounds that could become important. Yeah.
Do you know whether that would be part of the new version? Would there be some attention paid to changes in absorption? I guess this is the question perhaps for the SimSip folks. Will that be sort of within scope for your next iteration?
To be honest, if you had any examples of compounds that would have data for hepatic impairment which show this effect, then we could definitely consider it. I'm not aware in the literature at the moment, but Trevor may be able to comment further. But yeah, back to you if you knew any outside the scope of your work, or how many in your project were actually impacted in that way, do you think?
Yeah, I mean, Tycho seems to be on top of that subject, so I would defer to him. But I do think we do feel we have a few examples that we could bring to your attention.
Yeah, one thing is to understand whether for these lipophilic compounds, the bile solubilization is important for absorption. And that means we should be able to distinguish, to answer the question if we have solubility limited absorption, which might be a little tricky. because we do have quite few lipophilic compounds that are quite well absorbed anyway.
I'm gonna unmute Taicho, who'd like to ask a question.
Yes, I feel like I'm gagged. Okay. Um, so, so we know that certain compounds which have this very high positive food effect require bile acids to be solubilized. I think that's very well understood. There are compounds such as, telaprevir, for example, which show this. And it is true, there's not many compounds, but the issue is that it is not good if we do a prediction for a compound simply based on plasma protein binding and CYP3A4, because usually that AUC ratio will go up. But you have to remember that even if you have a food effect of 2,4, that means bile acid and bile acid composition requires or facilitates the absorption, which is completely not covered in the model, and therefore the prediction that we have could be overestimated, and that should be fixed. Minimally, what I would recommend is that modelers would have the option of somewhat reducing absorption in the model as sort of a, see what happens to the AUC ratio. If you can't include the bile acid reduction output in severe, particularly in severe impairment, right? I mean, there are some data out there that show that the bile acid concentration and composition is significantly altered. So for you guys, it should be easy to fix.
In terms of the architecture, yeah, it's there to be able to account for that.
I think you've got a comment from Masood as well, so I'll unmute Masood. Give me 1 second.
It might be easier.
Yeah. Hi, Masood.
Hi, thank you. I think Sheila and the group, this is very great work and lots of effort obviously have gone into it. I think one comment before I get to my question is that, as Oli said, the framework is there. So if we know how much the bonds that they are reducing, we can incorporate them in the population and then see the impact, as Oli was just explaining. So my question is to IQ group that now after this work, you have increased the confidence in the capability of the PBPK model to predict many of these scenarios. I don't know if any of the companies on the back of this work have started to be a bit more brave and submit simulations for organ impairment, so either renal or hepatic impairment. Has there been any changes that you start submit this to regulatory, and whether there has been any feedbacks on the back of this work?
We have just had some questions from EMA regarding this, and planning to use this result, this paper to show that we could do with prediction. So this is something, so we don't know yet. We just have received some questions, and we don't know the outcome, but yes, certainly we are, at least in our company, we are trying to do that.
And I think we've got another comment from Trevor.
Hi, yeah, can you hear me?
Yes.
I think it's a good comment actually on the bile solubilization. We've actually not done an awful lot on the absorption side with the cirrhosis population. So this is maybe something we can get together with our ADAM team and start looking at seriously. I just did a sort of quick search, and actually there is, as Tycho was sort of hinting, there is sort of fairly good evidence that bile salts are reduced in cirrhosis. So that is certainly one area we can sort of look at. The other thing to sort of emphasize as well, going on from Steve's comment, was that we've actually introduced the shunting model now into the minimal model from version 21. So whichever model you use, you'll be able to account for the effects of shunting as well. Another thing with our version 21 data, some of the early SIP data, we're actually reducing the sort of extent to which certain SIPs are being reduced in cirrhosis. So that may help the model performance as well. But this study has been extremely useful for us, and thank you for your presentation, which was extremely clear.
Yeah, thank you, Trevor.
If I could just go back to what Masood was asking, you know, are we using it? I think what Sheila's done a great job of here is getting away from the question, you know, does it work or not? Because if we just think like that, then we get stuck. But this totality of evidence and putting things in context and using the information without saying we're going to replace a study necessarily, but adding to the overall profile of the molecule. That's something that we are doing, you know, and it's an ongoing conversation that we have with the regulators. So I think that's a big step forward. And I hopefully the FDA and the EMA see it in the same light, that it's not, can you use this model or not? It's what's the context in which this model enables you to add value to a particular conversation. And that's where I think Sheila did a really good job of illustrating here today.
Thank you, Steve.
Thanks again, Sheila. I don't know if there's any other questions or comments, but it's been really great talk and really great discussion as well on the back of it.
I think there is one more comment from Dan Scotcher. I'll just unmute him.
Yeah, sure.
Oh, thanks. Great talk, Sheila. Thanks. I just had a question on the alternative staging of hepatic insufficiency that you have on this slide, actually. Is there anything that you're particularly looking at, any particular biomarkers that you're considering for this?
I don't know about this. Steve or Tycho, anybody can come in for the biomarkers?
Well, it's a very, very old question, right? Is there some magic biomarker? The diverse array of enzymes and transporter activities that in this disease. So I don't think that's going to happen, but it may be, if we said, do we have a CYP3A4 biomarker? Maybe there's a path forward there. So I think breaking that big question down into individual pathways, maybe we can be productive. As you know, there was a big FDA workshop about this at the end of last year, and I don't think we reached a conclusion other than there's a lot of activity and a lot of interest in that, but I don't know that we have a clear path.
What about the liquid biopsy work that is being done in University of Manchester and Capcar?
Yeah, what about that, Dan?
I'm not directly involved. I know that there's a lot of potential there for sure. I think it's a watch this space. I mean, I actually think the imaging could be quite interesting as well, because the imaging is already, you know, to some extent set up within the cirrhosis, you know, clinical space. But I guess that apart from experiments all the way to being used by everybody across the board, because you don't want to end up in a situation where it's only going to be one or two hospitals that have this biomarker, which is the risk I see potentially with the liquid biopsy and anything else that wouldn't be picked up by every hospital across the world. Like, you know, that's the value of creatinine really for the renal. Everybody uses it.
You've got a comment from Amin. Amin.
Can you hear me?
Yes.
Well, I agree with the last comment by Dan to start with, you know, because the AmpliSeq is becoming so common that in majority of the hospital centers, actually, they have it. Of course, translating the result is a different matter, and you need tools, to do that and say that, what it means. But going back, to the question, in fact, Steve honestly knows as much as I know, because he attended alongside me, to the focus group on the liquid biopsy current project that is happening. And by chance, not by design, we are apparently ending up on set of samples that it seems to be containing both renal impairment as well as hepatic impairment, because we couldn't actually source any other suitable samples to do the liquid biopsy. So the latest, this is basically, was it yesterday or the day before, Steve? I can't remember actually. But we had a meeting that it seems that these are the samples that they are going to be going through the next round of liquid biopsy. So hopefully in 6 months time, I can answer this question.
Good.
I think that's all the questions.
Thanks again, Sheila.
Thank you for the opportunity.
Yeah. Do you mind just putting up the last slide?
Yeah.
So just to highlight that exactly a month's time, we have our next talk in this series, and it's looking at bioequivalence. It's Yanis, who's currently in Novartis. He is talking about conducting virtual bioequivalence, focusing on biopharmaceutics. So please join us there. Register again in the same way. Thanks again, and take care. Till next time.
Yeah. Thank you very much.
Thank you.
Bye.
Bye.
Take care.
Bye, everyone.